Hello everyone, this is Jonabel Longos and let me share to you my topic about genes and chromosome structure. The common questions are, what are chromosomes? Where are they located in the cell? What are they made up of? So we have this simple cell as an example. If you can see the thread-like structure in the nucleus, these are what we call the chromatids. They are usually tangled when present in a regular active cell. When the cell separates these chromatid threads, the process is aided by condensation of threads. The condensation of threads is when the chromatid materials will gradually start coiling like a telephone wire. As a result, the thread will gain another structure. So this is the structure of a chromosome. Each cell has a pair of each kind of chromosome known as a homologous chromosome. Chromosomes are made up of chromatin, which contains a single molecule of DNA and associated proteins. Each chromosome contains hundreds and thousands of genes that can precisely code for several proteins in the cell. The structure of a chromosome can be best seen during the cell division. Human cell contains 46 chromosomes, one set of 23 chromosomes from the mother and another set of 23 chromosomes from the father. The chromosome contains hundreds to thousands of genes. So what are genes? Genes are the basic physical and functional unit of heredity. If we zoom inside the segment of the genes, we will find these molecules that make up the segment, the DNA molecules. So genes are contained in the chromosomes which are in the cell nucleus. An international research effort called the Human Genome Project, which worked to determine the sequence of the human genome and identify the genes that it contains, estimated that humans have between 20,000 and 25,000 genes. So this is the basic idea of the genetic materials in our cells which we need to understand. The chromosomes are made up of genes and the genes are made up of DNA. So what are the functions of the chromosomes? The main function of chromosomes is to carry the genetic material from one generation to another. It also protects the DNA from getting tangled and damaged. Each chromosome contains thousands of genes that precisely code for multiple proteins present in the body. The types of chromosomes depend upon its structures and its functions. So this is the structure of a typical chromosome and it is a single chromosome. The difference is that this single chromosome is not duplicated while the other one has a duplicated copy attached to itself. So these are the duplicated sister chromatids which are attached to the same chromosomes. And notice that the chromosome has its two arms. The P arm is the smaller one and the Q arm is the larger one. So this circle that attached to the center of the chromosome is what we call the centromere. This is the structure to which the arms are attached. It's the portion of the DNA that acts as a link between two sister chromatids, and its major function is the attachment of spindle fiber during the cell division process. It is also the position of the centromere that helps categorize the chromosomes into four types. The first chromosome is the metacentric type. The centromere is placed almost exactly at the center. The two arms seem of the same length. The next type is the submetacentric. Here, the centromere is placed slightly away from the center. As a result, the two arms of the chromosomes appear unequal. The next type is the acrocentric. In this, the centromere is almost towards the end region. The term acro in Greek means peak. Thus, the acrocentric refers to the type where the centromere is near the terminal region. Lastly, the centromere is present at the extreme end, and that is, the centromere is placed at the telomeric region. These telocentric chromosomes are not found in human. The human karyotype lacks telocentric chromosomes. So this is just a representative image of a human karyotype. The chromosomes that pair from 1 to 22 are called autosomes. Both contain genes that code for protein, which make up our body, and they are not the sex chromosomes. The last pair determines the sex of the person. If the two are both X and X, then it is a female individual. And if the one chromosome is X and the other one is Y, then it is a male. So this pair is the sex chromosomes. So this is the structure of a eukaryotic chromosomes. This is the single-stranded chromosomes that undergoes replication and becomes double-stranded chromosome. Take note that chromosomes are only visible when a cell is dividing, so we usually see them in their double-stranded form. Each species has a specific number of chromosomes. The two sets of chromosomes is what we call the diploid, which is found in all the non-sex cells or autosomes of an organism's body. And the other one is the haploid, which is one set of chromosomes. Only the sperm and egg cells have the haploid number. And this is also the structure of a prokaryotic chromosome. The prokaryotes have a one single loop of DNA. They don't have a true nucleus. So what are the difference between eukaryotic and prokaryotic chromosomes? So in terms of their shape, the eukaryotic chromosomes are linear, and for the prokaryotic, they are circular. And the size, the eukaryotic chromosomes are large and prokaryotic are small. And for the eukaryotes, the number of chromosomes are multiple, and prokaryotic are uh, there is only a single chromosome. And the location, for the location, the nucleus or the chromosome is located in the nucleus. And for the prokaryotic, they are located in the nucleoid, the region in cytoplasm. The storage proteins is the histones for the eukaryotic chromosome and nucleoid associated proteins for the prokaryotic chromosomes. Next is human karyotype. So in karyotype, it shows the complete set of human chromosomes. Each human cells contain 23 pairs of chromosomes. The sex chromosomes in males are the X and Y, and for females, the X and X. So these are the examples of karyotype for male and female. The human body has about 20,000 different genes in each cell. So each gene has a specific function. Now, when a gene or chromosome is abnormal, it may cause health problems in the body. So there are two main types of genetic changes: the chromosome abnormalities and the single gene defects. First, what are chromosome abnormalities? So it is a type of genetic disorders caused due to the change in one or many chromosomes or the abnormal arrangement of the chromosomes. The most common abnormalities is what we call the aneuploidy. It is a condition in which there is a loss or gain of numbers of chromosomes due to abnormal segregation of genes during the cell division. Examples of disorder include the Down syndrome, or what we call the trisomy 21. It is a type of trisomy as there is an extra copy of chromosome 21. If you look at the karyotype at the right, so we can see that there is an extra copy of chromosome 21. So it happens in 15 in every 10,000 births. So this is an example of a baby who is having a Down syndrome. The symptoms are the person is short and has a small and round head. The physical and mental development is retarded. It has furrowed tongue and partially open mouth and broad palm. Next is the Turner syndrome or monosomy X. One of the two sex chromosomes is not transferred. This leaves a single X chromosome for 45 total chromosomes instead of 46. So this happens 2 in 10,000 female births. These are the features for Turner syndrome: wide or web-like neck, low-set ears, broad chest with widely spaced nipples, high, narrow roof of the mouth, arms that turn outward at the elbows, the fingernails and toenails that are narrow and turned upward, swelling of the hands and feet, especially at birth. They have slightly smaller than average height at birth, slowed growth, short fingers and toes, and also infertility. Next is the Klinefelter syndrome. The genetic disorder arises due to the presence of an additional X chromosome in males, thus resulting in a chromosome count of 47 instead of 46. So this happens 10 in 10,000 male births. The features for Klinefelter syndrome include narrow shoulder, short trunk, smaller testicles, poor muscle development, enlarged breast. They have longer legs, and such individuals are sterile. Now let's go to the structural abnormalities of the chromosomes. It is when the large sections of DNA are missing from or added to a chromosome. The structural abnormalities can take several forms. Number one is the deletion. This is when part of a chromosome is missing or part of the DNA code is missing, just like what you have observed in the picture. The second structural abnormalities of the chromosome is the duplication. Some part of the chromosome is repeated, resulting in extra genetic material. Another abnormality is the inversion. It is when a chromosome breaks and the piece of it turns around and reattaches to itself. Inversions can be passed down in families, but they may or may not cause birth defects. Next is the translocation. It is when a chromosome segment rearranges from one location to another. It can happen either within the same chromosome or move to another chromosome. Translocation has two types, the reciprocal and Robertsonian. For the reciprocal, the segments from the two different chromosomes are exchanged, and for the Robertsonian, an entire chromosome attaches to another chromosome. Another structural abnormalities of the chromosome is the ring or a ring chromosome is when a portion of a chromosome has broken off and the ends are attached to itself to form a ring. Rings can be passed down in families. They may or may not cause health problems. Now let's have another types of genetic changes, which is the single gene defects. A change in a single gene causes a defect or abnormality. Single gene changes usually have a higher risk of being passed on to children. The gene changes can be dominant or recessive. For the dominant, abnormality occurs when only one of the genes from one parent is abnormal. If the parent has the disorder, the baby has a 1 in 2 chance of inheriting it. Examples include achondroplasia and Marfan syndrome. The achondroplasia is a bone development disorder that causes dwarfism, and the Marfan syndrome is a connective tissue disorder that causes long limbs and heart defects. The gene defects can also a recessive. For the recessive, the abnormality only occurs when both parents have a copy of an abnormal gene. If both parents are carriers, a baby has a 1 in 4 chance of having the disorder. Examples are the cystic fibrosis, the sickle cell disease, and Tay-Sachs disease. The cystic fibrosis is a disorder of the glands that causes excess mucus in the lungs. It also causes problems with how the pancreas works and with how food is absorbed. The sickle cell disease is a condition causes abnormal red blood cells that don't carry oxygen normally. And for the Tay-Sachs disease, it is an inherited condition that causes the central nervous system to decline. The condition is fatal and usually by age of 5. So how genetic changes are passed along in a family? The risk of having a baby with a birth defect from a genetic change increases if the parents have another child with a genetic disorder. There is a family history of a genetic disorder. One parent has a chromosome abnormality, and the baby in the womb has abnormalities seen on an ultrasound. So, for families at risk for genetic diseases, may want to talk with a certified genetic counselor. Making a chart of the family members and their health histories can help show risk for certain problems. Genetic counseling also helps parents understand the effects of a disorder and ways it may be prevented or treated. Also, getting genetic testing. Each parent's DNA may need to be checked. This is done to learn about some genetic inheritance patterns. So, prenatal testing can also be done to check the baby in the womb for problems. So, that would be all for my topic, genes and chromosome structure. Thank you so much for listening.